Again, we're going to do another signal transduction version of G protein coupled receptors initiated signal transduction, again, for hydrophilic hormones only, not for the lipophilics. Remember, the lipophilics only includes the thyroid hormones and the steroids. So this is a possibility for all other hydrophilic hormones. We did a previous video which was for adenylate cyclase and guanylate cyclase. So in other words, a cyclic AMP, cyclic GMP mediated signal transduction. And what I want to do now is swap out that enzyme for phospholipase C, because that acts in a little bit different, well, a lot of bit different way than the adenylate cyclase or guanylate cyclase pathway. So, same starting picture. Big cell. And our receptor, again, binding site faces the extracellular space, the ISF, because the hormone is hydrophilic, which can't enter the cell, which means it has to have a binding site that's exposed to the ISF. That's a receptor. This is the ISF. All right, here's our triangle-shaped hydrophilic hormone that finds this receptor, binds to it, and then initiates. Remember, these are recruiter receptors. The G protein coupled receptor is a recruiter receptor that recruits the G protein and the enzyme in question, which in this case will be phospholipase C. So we're going to add our G protein. Right, remember, it can be stimulatory or inhibitory to the action of the enzyme in question here, which is PLC, for phospholipase C. All right, so this enzyme, once activated through this sequence of events, is a lipase. Lipids, right? And lipids is another term for fats. So it's acting on a fatty substrate molecule. Well, fatty substrate molecules are going to be found associated with, well, one of the places they're associated with is the membrane, because the membrane has that fatty component, right? The fatty acid layer in the middle of the membrane. And so the I'm just going to draw an arrow here. The phospholipase C is going to act on that substrate, which is part of the membrane. Now the question is, where do you want me to write this substrate name? I don't want to write it on the outside of the cell. Maybe I'll shorten my arrow here a little bit. Right, but remember that it's not floating around in the cytoplasm either. Right, and it's PIP2. Okay, that is the lipid substrate for PLC, which again is found associated with the membrane. Right, PIP2 stands for phosphatidylinositol 4,5-bisphosphate. No, you won't need to remember that. But you need to identify, when you see PIP2, recognize it as the substrate for PLC. All right, well, PLC is an enzyme, and what it does is it cleaves PIP2 into two separate molecules. One of those molecules is going to similarly be lipophilic, and one of them is going to be hydrophilic. So the hydrophilic one will diffuse off into the cytoplasm because it's hydrophilic, and that one's called inositol triphosphate. Okay, the other one, IP3, the other substrate, that's the lipophilic one, right? This is hydrophilic. The other one is called diacylglycerol, or we're going to call it DAG. That guy, again, I'm going to write it here, sort of associated with the membrane still, because it's lipophilic. So now we have two second messengers, right? And those second messengers are going to go off and do their thing here. So we got to pick a direction and follow one of them. The IP3 is going to go off and act on actually going to bind a receptor, but it's going to act on an organelle called the endoplasmic reticulum. And the endoplasmic reticulum, it serves as a storage site for calcium, right? So it keeps the cytoplasmic calcium levels low by sort of pumping it into the ER and storing it here. So the IP3 is actually going to bind to and open an IP3 dependent calcium channel. So our calcium concentration, it's a divalent cation, right? So it's 2+. So our calcium ion, once we open this channel, will diffuse out and increase cytoplasmic calcium levels. Well, as you've seen before, cytoplasmic calcium is like, it's like the little kid that runs around the house turning on all the lights and, you know, opening all the faucets, right? Calcium turns on all kinds of things inside cells. In muscle tissue, for instance, calcium is the initiator of contraction. In neurons, it's the initiator of neurotransmitter release. In endocrine cells, it's the initiator of hormone release, and on, right? And so generally we keep cytoplasmic calcium levels low unless we want to activate whatever that rise in calcium is intended to do in that cell. Okay, so we've got a rise in calcium. Now let's go over to the DAG for a second. The DAG acts on a kinase called PKC, right? K for kinase, right? And that kinase, of course, can, right, that's what kinases do, they phosphorylate, right? So that kinase can phosphorylate, so I'm just going to have protein phosphorylation in that room. And I'm just going to say change function, right? Because remember, the phosphorylated proteins can be enzymes and channels and transcription factors and pumps and all those things that we can change the activity of by adding the phosphate group. Okay. Now, and one of the things that the PKC phosphorylates is a calcium channel. Put it down here, right? So we have a calcium channel, and it's got a gate on it, and we're going to show it's open now. And we're going to show a little phosphate group stuck to it because we phosphorylated it, right? So here's our channel. There's the gate on it, and that gate swung open once it became phosphorylated. So now guess what? Well, you have to remember we have high extracellular calcium levels and low. Yes, we're letting some out here, but remember it's 10 to 20,000 fold difference in concentration of calcium between inside here and in the cytoplasm. The same is true here. So calcium will still enter, right? We add more calcium from outside the cell. Okay, calcium went up again. We may have altered the function of other proteins, right? But now we have this rise in intracellular calcium. Well, calcium binds to a calcium binding protein called calmodulin. I'm going to run out of room here. So my calmodulin, I'm going to put these calcium molecules, I think it binds 4, but it doesn't matter. I'm just going to put a couple on here. Right? That calmodulin binds to the free calcium and becomes activated. Well, the calmodulin can now, right, and this is a protein that's binding these ions, and that protein, guess what? There are CAM-Ks. In other words, calmodulin-dependent kinases. Remember when we did cyclic AMP or cyclic GMP, we had CAMP-Ks, right? Cyclic AMP-dependent kinases. Well, now we have calmodulin-dependent kinases. And I'm running out of room here, so I'm just going to say activates kinases, right? And we get what we talked about before, protein phosphorylation, right? I'll add my phosphate group on it here. And remember those phosphorylated proteins, we've just altered the function of. Okay, so now we have a change in function of this cell mediated through activation of kinases directly through DAG, or activation of kinases through the calcium calmodulin dependent active action here, right? Different set of kinases that become active, and we phosphorylate probably different proteins. All right, so this cell is now activated, okay? Let's follow through though, remember, because before we talked about when we did the cyclic AMP, cyclic GMP, we had to talk about how we would turn off this hormonal response. And so again, we have to do some of the same things. We have to get rid of the hormone, so we would enzymatically degrade the hormone. So enzyme degrades right? And again, that doesn't mean this cell will stop responding because the enzyme had been activated, and again, assuming GS protein, the enzyme had been activated, we got cleavage of the PIP2, we have our second messengers, well, they're still there. So, and I'm not going to try and squeeze in all the terminology on here, the labels on here, but we would enzymatically degrade the IP3 and the DAG, have to get rid of the second messenger. Are we done? No, because we already phosphorylated over here, right? So we have to dephosphorylate. What's the enzyme that does the opposite of a kinase? Phosphatase, right? So we would dephosphorylate through a phosphatase action, right? Including the channel that we opened. So again, if we dephosphorylate this with a phosphatase, that channel would close. And intracellular calcium levels would no longer rise. If we get rid of the IP3, this channel would close, and we would no longer be releasing calcium. Well, does that mean the calcium levels in the cytoplasm go back down? Not without something else, right? We have to get rid of the calcium that we released so that we could deactivate the calmodulin. And the way we do that is through one mechanism you've seen before when we laid out how cells set up their electrochemical gradients. And that's through the action of, squeezing it down here. There's a calcium ATPase that just pumps calcium out of the cell all day long, tries to keep the intracellular calcium low, has to be overwhelmed by other systems to get that calcium level rise. So this guy's going to pump out the calcium, right, back to where it came from. And we also have a pump, let's see, I'll also make it green, pumping calcium into the ER. That's called the SERCA pump. I don't really have a place to put that. SERCA, S-E-R-C-A, right? Smooth endoplasmic reticulum calcium ATPase, SERCA. And that pump sits here and pumps calcium into the ER all day. Again, trying to keep the concentration in here high and the concentration down here low. So once these two remove the calcium, it's going to diffuse off our calmodulin, deactivating it, which means we're no longer going to activate our kinases. And we have to take the last step, of course, which was those kinases were already active for a little while, which means they phosphorylated proteins, which means we need to dephosphorylate those proteins, just as we did over here, right, through the PKC pathway. And so our phosphatases would act on to dephosphorylate these proteins, reversing the initial action. Okay? Interestingly, for any of these signal transduction mechanisms that activate kinases, there may be alternative ones. And again, we're giving you the majority response here, but there are others that are actually linked to phosphatase activity instead of kinase activity, which means they would tend to do the exact opposite of this scenario. In other words, they would tend to dephosphorylate as their action, as opposed to phosphorylate as their primary action. Right? Okay, so I think we've got our hydrophilic hormone signal transduction mechanisms, right? We did adenylate cyclase and guanylate cyclase in a previous video, and now we've done the PLC and the IP3 DAG, right? We still get amplification because we get lots of this. You could think of these two as being second messengers. You could also think of calcium being a second messenger. That's fine. But we get amplification, we get activation of kinases, and we've changed cell function by altering the types, amounts, or activities, depending upon which proteins we're talking about here, to alter cellular function, right? Types, amounts, or activities of cellular proteins. Okay.